Yep. So my name is Sammy Lee. I'm the Director of Business Development at Biodesign Lab. We're a synthetic biology company based in Seoul. What's the problem we're trying to solve? Gene and cell therapies are advanced therapies that hold immense promise for treating a wide range of diseases. And as you can see by the market forecast here, the market of gene and cell therapies are predicted to dramatically increase and expected to reach over $80 billion by 2027. And because a majority of gene and cell therapies commonly use a tool called viral vectors to deliver the genetic materials to the cells of interest, as the market for gene and cell therapies grow, so does demand and the supply for viral vectors. However, the current generation of viral vectors have limitations affecting the safety and efficiency, both of which can increase the risk and cost to patients. What I mean by safety is that viruses by nature want to increase its chances to replicate, so insertion of the genetic cargo that it's carrying are frequently offloaded at regions that cause detrimental effects. Recent clinical trials have had setbacks caused by these risks to safety. What this means is effectively there are delays in the treatment approvals, leading to more costly research and more longer clinical trials. On top of this, current viral vectors are not very efficient at gene delivery efficiency. So there's a growing need for safer viral vectors with more efficient delivery of the gene, and this will reduce the risk and development costs and also reduce the time to get the therapies onto the market. So how are we doing this? How are we solving this problem? The root cause of this problem is the viral vectors that they're based upon have high tendency to insert the genetic cargo at gene regions such as regulatory regions and gene regions. So as you can imagine, inserting at these tumor suppressing region or a pro-oncogenic region would have severe detrimental effects, including the formation of cancer. Researchers have studied this for decades and have found methods to mitigate and shift the integration patterns away from these dangerous regions to improve the safety. However, this comes at a cost of gene delivery efficiency. This reduction in efficiency can be as high as 100-fold, making the vector impractical for therapeutic use. So Biodesign Lab is addressing these challenges with our innovative synthetic biology platform. We have developed RetroNex based on retroviruses, LentiNex based on lentiviruses, and they're built upon the current latest generation of viral vectors, and we've engineered them to be at least 5-fold more efficient at delivering to the genetic cargo to the intended cells, and also shifting the integration away from the dangerous regions that I mentioned previously, therefore reducing the detrimental side effects. We're doing this by using synthetic biology. A typical viral vector has about 10 components. Each of these components can be further biologically engineered and enhanced to give advanced features. So like a car assembly line, we can pick the right components that synergize for each for a specific application, and then assemble these components together to create an appropriate vector for that purpose. So right now we have about 6,000 components in our library, and we're constantly expanding our library for future applications. Comparing our vector to the ones on the market right now, we have multiple advantageous features, as well as the safety and efficiency that I mentioned. But what it all boils down to is that we can make new gene and cell therapies a lot more safer, a lot more efficient with lower doses, and faster to the market. And this all means that a new therapy can be lower cost to the end patient. And the team behind this is Dr. Choi, who's from University of Cambridge, who brings the computational side of the technologies, and Dr. Lim, who found the key component while at UC Berkeley that shifts the integration and insertion of the genetic cargo away from the dangerous regions to produce safer vectors. Our business model, we are looking to license out to pharmaceuticals and biotechs. We are currently partnered with domestic companies such as Celltrion GC Cell. We've only just started advertising ourselves March last year, and we've garnered some global interest from partners. And we are currently in Series B and wish to use these funds to grow manpower across the board while developing more strategic partnerships. We also wish to eventually manufacture our viral vectors ourselves and produce therapies ourselves with our viral vectors. But the main goal is to grow more strategic partnerships with global companies. Thank you.